All right, we'll see. I might get rid of this today and just project onto the wall. I never know if I've got this plugged in, if I have two USB plugs, if it's going to mess it up, so I have to come over here and keep clicking. So everything we're talking about in this PowerPoint is all about metabolism, so all the different chemical reactions that are happening inside of the cell. One of the big things that helps all of these chemical reactions happen are the use of enzymes. I was going to say they don't always speed up a reaction, but they kind of do because it's more likely to happen if enzymes are present. And so I left off, I was talking, I had my little frogs come up here too. If an enzyme is present, it lowers how much energy is needed to get a reaction going. Reactions don't happen spontaneously. They need a little bit of energy just to get the reaction going. No matter what kind of reaction, even if we get energy out of it, we still need some energy to go in just to start it all off. And enzymes just lower how much energy is needed. Less energy is needed, so this is how much energy is needed without an enzyme. This is how much energy is needed with an enzyme. That frog, you know, it's a lot easier to jump over a wall if it's a lot shorter. So it just lowers the amount of energy that's needed. Couple more things about enzymes. One, enzymes are proteins. And so again, we need proteins in our diet for lots of different reasons, and part of it is because enzymes themselves are proteins. Now, enzymes in most textbooks, enzymes are represented as big blue or purple blobs. I don't know, I swear all the textbook people company got together and like, let's always draw them as big blue and purple blobs. But they do all have an active site on the enzyme. And an active site is where the substrate is going to bind. So an active site is to bind to a substrate. And it is very specific for what substrate can bind in that active site. Now what ends up happening is after a substrate, which is very specific for the enzyme that will bind to it, once it fits into that active site, it does form more of a perfect fit. So if you notice, this substrate will fit here. However, this is like a perfect lines where this is more of a curve, but the substrate will fit there. It's a very specific substrate which will fit there. And then once it fits in there, it forms a more perfect fit. Now, a couple more things about enzymes. One, I thought I brought one up that had all this written in. Enzymes are not used up or changed. That's how the first line's on there. So enzymes are not used up or changed. They are recycled. They are specific for one substrate. And they drive a reaction one way. Now, when they drive a reaction one way, they're also known as, which is the last line, a catalyst. If you want the reaction to go the other way, you would need a completely different enzyme. So on this picture, because it just looks like a lot on there, you don't have to know anything about the picture, but it's an example. So big blue blob, so it's an enzyme. And this particular enzyme's name is fructose 1,6-biphosphate aldolase. It's like a mouthful. Most enzymes do end in ase. It's like a big clue that it's an enzyme. And the name of the enzyme is a clue on what the substrate is that can fit in there. Again, they're very specific. So if the enzyme's name is fructose 1,6-biphosphate aldolase, it's because the substrate that fits into its active site is called fructose 1,6-biphosphate. So the enzyme name itself is based on what the substrate is that fits into it. And a lot of enzymes are named that way. Not all, but a lot are. But we can see that that substrate, that fructose up there, fits into the active site. There's the enzyme. It's got a more perfect shape now. And the job of this particular enzyme is to break that fructose into two different things. Once it's done that, it's made the products, those two different molecules, the enzyme is right there being recycled to go find another fructose 1,6-biphosphate and do it all over again. So it's recycled and it's very specific for what it does. And so it just keeps getting used over and over and over again as long as the substrate is present. It will make the products. Now there are things that can change how well enzymes work. One of the big ones is temperature, because if something gets too hot, enzymes can denature. The pH can interact with some of the bonding, and enzymes won't work very well. How much enzyme is present compared to how much substrate is present depends on how well that enzyme is going to work. If there's a lot of enzymes and a lot of substrate, it's going to sit there and work very, very quickly. If you have an enzyme and not a lot of substrate, you got a lot of that fructose aldolase, but not a lot of fructose, I'm like, the enzyme's not going to do a lot if it doesn't have a lot of substrate. Because again, we'll only have that one substrate that can fit there. And whether there are inhibitors present. And there's a couple different types of inhibitors. Well, most of their job is to block the active site. I'm going to close this door. I think it's unlocked. Because again, the active site is where that substrate binds to. And if we change that, substrate can't bind. Here's my first couple things that can change the enzyme activity. Again, most enzymes have an optimal temperature they work under, a pH they work under, how much substrate is present. And this is just showing a couple different examples. Most of our enzymes work best at body temperature. However, there are organisms out there that live in hot springs that their enzymes would work better at more of closer to a boiling temperature, at least a hot temperature. So a couple examples of inhibitors. We group them into competitive inhibitors and non-competitive inhibitors. Either way, they both inhibit. If it's a competitive inhibitor, it means it is something that is going to compete for the active site. So one specific substrate can fit in that active site. But could something else fit in there and then block the substrate? Yes, and that's what a competitive inhibitor is. It's competing for that active site. So here's your big blue blob. It's an enzyme. And its job, its active site, is to fit this substrate, which should fit in that active site perfect. However, this little itty-bitty inhibitor was like, well, I can kind of sneak my way in there and fit into that active site. And if it's sitting there, the substrate can't bind in the active site. And so if an inhibitor is present, it kind of takes over and they both compete for sitting in that active site, which is going to decrease how much substrate is reacted with. And so if substrate can't bind, you don't get any products. This is just showing an example. This is the substrate that's going to fit in this particular active site. But if this little inhibitor is around, it's going to fit in there first. And now the substrate can't bind because it's already busy. A non-competitive inhibitor. It's still going to inhibit, but it is not competing for the active site. So it's still some other type of molecule that's going to bind somewhere else on the enzyme. There's usually another spot on an enzyme where other things can bind. It's not the active site. It's usually called an allosteric site. And the inhibitor can bind somewhere else. And when it binds into that other site, the enzyme changes shape. Just enough that that active site changes a little bit. And so the substrate can't bind. And so I'm going to go back. It's the same enzyme. If you notice, there's that allosteric site. This is the substrate. It should fit right here in the active site on that enzyme. But if an inhibitor binds down here, it causes that enzyme to change. All of a sudden, it curled over a little bit at the top. And because of that curl, that substrate can't bind. And so again, it's an inhibitor because the substrate can't bind. The only big difference is it's not competing for the active site. So the inhibitor's not trying to bind into the active site where the substrate normally would. It binds somewhere else. It binds in that other alternative allosteric site. This is showing another example. Here's where it normally should bind, your substrate. But if that inhibitor binds in that other site, it causes the enzyme to change shape. Now that substrate can't fit in. You're not going to get any product formed. So if inhibitors are around, they're going to decrease how much product is made. Now, everyone's like, oh, okay, so inhibitors are bad. Well, they are. However, sometimes our body will make inhibitors when we don't want to make any more products. It's called feedback inhibition. It's when our body will make an inhibitor so that we can stop making product. Again, our bodies want to try to be as lazy, efficient as possible. And you don't want to make product if you don't need that product. And so what can happen is, this is showing an example of different enzymes to eventually make an end product. So that particular enzyme only, in its active site, only fits little orange circles, and it's going to spit out little yellow triangles. That's its product. The next enzyme can only fit little yellow triangles. Its job is to then spit out little red squares. The last enzyme, the only thing that can fit in the active site are little red squares, and its job is to spit out little purple ovals, or little green ovals. However, what can happen is, when we've made enough end product, whatever this green oval is supposed to represent, and we don't want to make any more of that, sometimes the end products become inhibitors. And so that end product can actually go back up and fit in that allosteric site of the first enzyme, which causes that first enzyme to change shape, and now that little orange circle can't fit in, and then you don't make any more of the end product. It's almost like we need to make a product until we don't need to make a product. And so sometimes products can actually become inhibitors. So our bodies want to make whatever we need to make, but then we've made enough of it, they can actually become the inhibitor. Now, I'm gonna bring my screen up because I'm gonna write more on the board, more than just that little tiny area. We're gonna switch gears away from enzymes. So enzymes are gonna help all the reactions, and you'll see on some of my pictures enzyme names all over. But we're gonna talk about metabolism. Advanced A&P, have you guys gotten to carbohydrate metabolism yet? Okay. You will. This sheet is just for you guys to write all over. I'm not collecting it. Sometimes it's just nice to help organize notes when it comes to breaking down carbohydrates. Let me know if I counted wrong on there. Felt like they were two stuck together. Maybe there wasn't, and I miscounted. Again, you can write all over this. I am not collecting it. It's nice, though, because I am going to write all over the board and draw pictures today. The back side of it's a nice white sheet of paper. So if you want to write all over it, you can. Good, I counted right. There's a reason I got lots of colors. So carbohydrate catabolism, do you remember what catabolism means? This was last class. Yeah, breaking down. And so a carbohydrate catabolism means we're going to break down carbohydrates. Now, anytime you break a bond, you are going to release energy. And so in carbohydrate catabolism, we are going to be breaking lots of bonds, releasing lots of energy. This is how you guys get the energy to function during the day, is you eat stuff, you break it down, you release energy, and then you can go about and do whatever it is that your body needs to do throughout the day until you got to eat again. Now, when we have oxidation, we're talking about electrons. Do you remember there was oxidation? I'm not going to write out the word. And reduction. What's happening in oxidation? I'll write it out. Yes, and I'm like, so we're going to lose electrons. Why I care about electrons is electrons, because they carry that negative charge. Because they are charged, they do carry energy with it. And so when we're breaking things down and I say we were releasing energy, a lot of that energy is in the form of electrons. And so we care today what happens to the electrons and where do they go. Now, we're going to break apart glucose today. That's what most of metabolism is, all breaking down glucose. And glucose can get broken down in two processes, and we're going to focus mostly on this first process, the cellular respiration. Now, there are two parts to cellular respiration. Some of cellular respiration is happening if there is oxygen present, and some of it is happening anaerobically, where no oxygen is needed. And then depending on how far we get today, whatever I don't get through, I'll tape up in my office after the second lab. We'll see if we can get to fermentation. which happens all anaerobically or without oxygen. So this diagram, which has a lot on it, so we're going to break it apart into different steps. We're going to take, and I'm going to write all this out. We are going to take our first molecule that starts right at the top, our glucose molecule. We are going to break it down and make something called pyruvic acid. Again, I'll go through each step. We're going to take some pyruvic acid, we're going to break it down into acetyl-CoA. Acetyl-CoA is going to go into the Krebs cycle, and ultimately we're going to go to the electron transport chain. However, glucose, that's all cellular respiration, all the green. Glucose can also go over and do fermentation as well. So we're going to spend most of the time on the green, the cellular respiration breakdown of glucose, and then time dependent, we'll do a little bit of the fermentation as well. Now everyone's like, oh my God, it's like the worst picture ever. Yeah, I get it. First, do you guys know or remember from some science class somewhere what the formula for glucose is? Or any part of it? C6, H12, O6. Any carbohydrate, any and all carbohydrates always do have a ratio of 1 to 2 to 1. There's always 1 carbon to 2 hydrogen to 1 oxygen. Glucose, C6, H12O6, it's a carbohydrate. Now when we break apart glucose, it's a process called glycolysis. Lysis means to break apart. The glyc part of the glycolysis means the glucose. So glycolysis truly just means breaking apart glucose. And we're going to follow the 6 carbons. And so on the book picture, they do have all the little carbons represented as little black circles, and that's what I'm going to draw them out as. Now, if you're going to draw what I'm going to draw, I'm going to get colorful, just FYI. If you're using your sheet of paper, hold it this way and draw 6 little circles over on this far left side, because I'm probably going to use up a good chunk of that back side of the piece of paper if you're going to draw this. So like, put it the long way. Man, none of my stuff wants to erase well today. So, a couple of things that are going on in glycolysis. The word means to break apart, but what is needed to start any chemical reaction? That will help speed it up. That is the enzyme. So the enzymes lower the amount of what needed? The amount of activation energy. So energy does have to go in. Luckily, there are enzymes present, so not a lot of energy has to go in, but some energy has to go in. And I know there's a lot going on this picture, but at the very, very top, I don't think this can get any taller, way at the top, there are 2 molecules of ATP going in. Way up at the top on the right-hand side, ATP goes in, ADP comes out in 2 different places. So the energy that is going in to start this whole thing off, let's see what color do I want, use orange, is ATP. So I'm going to represent, my little arrows. I have 2 ATP going in to start the whole thing off. That's your activation energy. Now, there are lots of little steps along the way. We're not going to go through all of those. If you ever want to take a cell biology class, which literally the entire semester is all about the cell, you can learn all the little steps. That's not this class. So we're going to present lots of things are going on, and I care mostly today about the product, and then what else do we get out along the way. Our product at the very end is we get 2 pyruvic acid. Each pyruvic acid is a 3-carbon molecule. So at the end of glycolysis, I have 2 different 3-carbon molecules called pyruvic acid. But we got some stuff through all of these. We got some stuff out of it. Again, in glycolysis, I'm breaking bonds. I'm releasing energy. And so we're going to get some energy out. On here, I know there's a big line that goes through it. We're going to get 2 ATP out here, and we're going to get another 2 ATP out down here. So through all of those little steps, we have gained, so I got my arrow coming out, 4 ATP. So 2 ATP went in because we needed some activation energy. But I got out 4 ATP, so I'm ahead. But I got some other stuff out too. One other thing that I got out is I got out 2 NADHs. Now, do you remember why I care about NADH? It's like earlier on in this PowerPoint. Because I care about NADHs. What is NADH? It was one of 3 something. An electron carrier. So out of this, I'm going to get 2 NADH. Again, it's carrying electrons, which means it is carrying energy. So yes, I've got extra ATP, but I also have a couple of electron carriers. I'm also going to get a couple water out. I don't really care too much about the water. Part of the waters that are coming out are some of my extra hydrogens and oxygens. I don't care too much about them. But I do get them out. I don't want to go there yet. Now, on the sheet I gave you, things to fill out on the front side of where does all of this take place? And again, just the glycolysis. The glycolysis is taking place in the cytoplasm. And it doesn't matter what kind of cell. Our cells have cytoplasm, prokaryotic cells, the bacteria, they have cytoplasm. Glycolysis happens inside the cytoplasm. So for what goes in, there are 2 things that go in. One of them is 2 ATP. So 2 ATP go into the glycolysis reaction. The other thing that goes in is the glucose. Got to have glucose to go into glycolysis. For what comes out, we're going to have a lot more. You're going to have coming out 2 waters, 4 ATP, 2 NADHs, and 2 pyruvic acid. So you got 4 things coming out of glycolysis, all 4 of these guys. The blue, the orange, the red, and the black. For the box that it says energy produced, did I produce energy? 3 dots. Yes. How much energy have I really gotten out of this reaction? 2. I've only gained 2 ATP out of this whole reaction because it took 2 to make 4. So for how much energy have I gained? I've gained 2 ATP. So yes, I produced some energy in this reaction in the form of 2 ATP. For the anything else important, just by note, glycolysis, this step of taking glucose, and yes, lots of arrows, lots of things happening, and ending up with the 2-pyruvic acid. That glycolysis is part of cellular respiration and it is part of fermentation. So for the anything else important, that's the most important part. Glycolysis is part of cellular respiration and is part of fermentation. So it doesn't matter which direction you go, you still have to have glycolysis happening. So it's part of cellular respiration and it's part of fermentation. And so for that last box, is a part of fermentation aerobic or anaerobic respiration? It is part of all of it. So all of the above, or all. But we're going to follow it through the cellular respiration route. And so once I have pyruvic acid, I'm going to make some acetyl-CoA, go into the Krebs cycle, and then I know it's right on my line. I'm going to go into the electron transport chain. So I still have 3 things that are going to happen to my pyruvic acid. Oh, there's my 1, 2, 3. So we're going to take your pyruvic acid. It is also called pyruvate. It's got 2 names. And need my black. I'm only going to follow one of my pyruvic acid through. Just note whatever I draw is also happening to the other one. It's going to undergo some things. In my end, I'm going to have a 2-carbon molecule called acetyl-CoA. I think it's usually lowercase o. For acetyl coenzyme A. Now, what do I get out? What's coming out of the reaction? The NADH. How many? Just 1. However, my note, I'm going to get 1 NADH out of this pyruvic acid. I'm going to get 1 out of this one as well. I'm only going to follow 1 through. So we're going to total everything up because everything that's going to happen to this is happening to this, and it's all coming from 1 single glucose. So ultimately, my 2 pyruvic acid are going to net me 2 NADH. 1 here and 1 down there that I'm not going to draw, or my board's going to just look way more confusing. What else do I get out? It's the only other arrow coming out. CO2. I get some CO2. The only reason I really care about CO2 is I went from a 3-carbon molecule to a 2-carbon molecule. That carbon had to go somewhere, and that's where it is. It leaves as carbon dioxide. So to go from a 3-carbon molecule to a 2-carbon, the carbon had to go somewhere. It's not, just magic. So for the worksheet, where does this take place? The making of acetyl-CoA also takes place in the cytoplasm. And so again, it doesn't care if it's a prokaryotic cell or a eukaryotic cell. They both have cytoplasm. For what goes in, the only main thing that goes in is the pyruvate or the pyruvic acid. Whichever one you want to call it, it's the same thing. So just the pyruvic acid in. 2 pyruvic acids do go in. Because you do have 2. I'm only going to follow 1 through in my diagram. But 2 pyruvic acids do go in. What we get out are 2 NADHs, 1 from each pyruvic acid. So we get 2 NADHs. How many carbon dioxides? 2. And I'm like 1 here, I know I didn't write it in, and 1 here. Again, we will end up with 2 pyruvic acids will ultimately become 2 acetyl-CoAs. So the third thing that comes out are 2 acetyl-CoAs. Again, I'm only drawing 1 to make it less confusing. So you get 3 things out. 2 NADHs, 1 for each reaction. 2 carbon dioxides, 1 for each reaction. And 2 acetyl-CoAs, 1 for each reaction. Are we getting any ATP energy out? Nope, none. No ATP is made at this point. For, anything else important or other information, just that acetyl-CoA is going to feed into the Krebs cycle. So your ending product, that acetyl-CoA, is going to go to the Krebs cycle. Is it part of the fermentation, aerobic or anaerobic respiration? It is part of aerobic or anaerobic respiration. Most of the time this is done aerobically. This is going to be one I'm like, it is mostly done aerobically. Because you guys are aerobic, so any of this is all happening to you just like it's happening in bacteria. You guys are aerobic organisms. Most bacteria are aerobic as well, but there are some anaerobic bacteria that can do this too. It's mostly aerobic, but it can do it aerobic and anaerobic. So now we're going to head into the Krebs cycle. Again, lots of stuff going on. I don't care about a lot of it. So acetyl-CoA, I just made it. Again, there are two of them. I'm only going to follow the one through. And there's your molecule way at the top there called acetyl-CoA. That's your 2-carbon molecule attached to a coenzyme A, and it's going to go in. Now, that 2-carbon molecule is going to bind up with a 4-carbon molecule. There's your 4 little circles, the 4 carbons. I don't care that you know the name, but I'm going to say it as oxaloacetate or oxaloacetic acid. But a 2-carbon molecule plus a 6-carbon molecule is going to make how many carbons? 2 plus 4? 6. So you're going to have a 2-carbon molecule. It's going to bind up with a 4-carbon molecule. And now you're right back to a 6-carbon molecule. It's this 6-carbon molecule that is called citric acid, which is why sometimes the Krebs cycle is also called the citric acid cycle. Now, that 6-carbon molecule is going to go through a lot of stuff. I don't care about all of the intermediate stuff. But ultimately, you're going to go from a 6-carbon molecule right back to that same 4-carbon molecule. It's called the cycle for a reason. It's called the Krebs cycle because it just keeps cycling. I care about, as it goes from that 6-carbon citric acid molecule back to that 4-carbon oxaloacetic acid, what do I gain out of it? So we're going to look on here. What are some things that are coming out? What am I gaining? I'm gaining ATP, and how many? How many? 2 total, yes. So 1 here for this acetyl-CoA, and you'll gain another one because you'd actually have 2 acetyl-CoAs. So ultimately, at the end of the Krebs cycle, 1 glucose molecule will give you 2 ATP. 1 from each Krebs cycle, total of 2. What else do I get out? NADH, yes, and how many? Yep, so 3 on each Krebs cycle for a total of 6 because there's 1 there, and then 1 way up there. So ultimately, we're going to get 6 NADH. 3 from each Krebs cycle. Again, all from the same 1 glucose molecule. And there's something else. Water's going to go in. Again, it helps break stuff apart, which I'm not too worried about. But what else do I get out? Something else that's got an arrow coming out. Mm-hmm.
So
yep.
Why do we get like 2 of everything in the Krebs cycle?
Because
it goes around 1 time.
Yep. So this Krebs cycle is going to go around and give you 1 of everything, or 3. But I'm counting up everything for my 1 glucose molecule. So I made 2 pyruvic acids. I have 2 acetyl-CoAs. They're really, without making my drawing crazy, I would have 2 Krebs cycles. Does that make sense?
Yeah.
Okay. So that's why I'm doubling everything. Because there's another one, I'm just not going to draw it. I heard it though. The other thing we get out is FADH2. Why do I care about FADH2? What is it? It's another electron carrier. And how many FADH2s do I get out? 2 total. And I was like, 1 for each Krebs cycle. So that's the only place it's listed on here. 2. There's one more thing I get out though. And it's harder because it's not quite written out. You get CO2, thank you. How many CO2s? Without even trying to count up there. 4 altogether. Again, we go from a 6-carbon molecule back to a 4-carbon molecule. So 2 CO2s for each Krebs cycle. For 4 CO2s. And here's the CO2s. There's one of the CO2s that comes out. There's another one of the CO2s that gets lost. That makes us go back to that 4-carbon molecule. How many carbons? We started off with a 6-carbon molecule. How many carbons have we lost at this point? More than that. I started with 6. How many carbons have we lost? There's 2 there. 4 there. We've lost all of them at this point. Ultimately, at the end of the Krebs cycle, glucose is gone. And I'm like, it has been completely broken down, and you've lost all of that carbon as carbon dioxide. So that's when we end into the last step. And when I really care about all of those electron carriers that we've made along the way. Because the next step is called the electron transport chain. But I want to make sure you guys get stuff filled in on your thing. I think I've got another thing, you know. See, worksheet. Where does it take place? Any guess? Cytoplasm in one kind of organism. This is where it gets a little different. So this takes place, I shouldn't have written so far down. This takes place in the cytoplasm in prokaryotes. But in eukaryotes, can I spell today? No, I can't spell today. Eukaryotes. What organelle is the big ATP maker? Mitochondria. And this would actually take place in the matrix, hence my matrix reference up there, the mitochondria. But again, a prokaryotic cell doesn't have a mitochondria. That's way too advanced of an organelle. So if you're a bacteria, this would happen in the cytoplasm. Because then if you're taking advanced, or maybe you already took advanced, you're going to learn it and be like, no, this happens in the mitochondria. It does, because you guys are eukaryotic cells. But prokaryotes, they don't have that. Happens in the cytoplasm. What goes in? Well, I was going to say 2-acetyl-CoA. Because you have your you have 2 of them. I just didn't draw both. But 2-acetyl-CoAs are going to go in. And water. Again, I'm not too caring that much about water. It helps break things down, but water goes in. What comes out, though, is quite a bit. I think there's 4 things that are going to come out. You're going to get 2. I was going to say it's all my colorful things. And I'm like 2 ATP, 2 FADH2s, 6 NADHs, and 4 CO2s. It's like my 4 colorful things down on the bottom there. Did we get energy out of this? Yep, we made 2 ATP. Still not a lot of ATP, but we did get some ATP out. For my anything else important, I'm like, I wrote up here. You can shorten it if you want to write it on there. You can say, see PowerPoint slide. My only big notes for the anything else important is it's in this step that we get quite a few NADHs and we get now FADH2s. We've generated a good number of electron carriers in the Krebs cycle, and that becomes very important for our next step. We also do make some precursor molecules that can be used elsewhere. Again, we're not going through all the names, but we do make a lot of random little molecules. They all have a name as we went from that 6-carbon to a 4-carbon molecule. And these random little things, even some of the stuff down here, the GTP and stuff, it can be used elsewhere to build as building blocks. I just don't care about all of the named steps along the way. Again, if you ever want to learn more cell biology. Now, the electron transport chain, I'm going to try to draw this out and I'll come back to this picture. Oh, good question. And Mike, is it anaerobic or the aerobic? It is aerobic for aerobic organisms. And anaerobic organisms can do this as well. So it is mostly aerobic, kind of like the making of acetyl-CoA. It's mostly aerobic, but it can take place anaerobically as well for anaerobic organisms. You guys as humans are not anaerobic, so this would all take place aerobically in you guys. Now, like some of the book pictures for the electron transport chain look like this, and I'm like, that's the most horrible. Even myself would have to sit there and take like 5 hours to figure that out. I don't like it. So I found this from a different textbook. That looks more doable. And I'm going to try to draw this. Again, I am not artistic whatsoever. And I'm not going to draw all of those. So big blue blob. If it's a big blue blob, what is it? Some type of enzyme. Here's your phospholipid membrane. I'm not counting up all the tails, I don't care. Phospholipid membrane. Now, I'm actually even going to start before I start drawing anything else out. For where does it take place? Based on the picture at the top, where does this take place? Mitochondria. And really, I'm like, on the inner membrane of the mitochondria. The inside of the mitochondria has all these inner folds. That's all phospholipid bilayer folds. So this is going to take place on the inner membrane of the mitochondria in eukaryotic cells. So this membrane is this inner fold, phospholipid bilayer. So it's the inner membrane of the mitochondria in eukaryotic cells. Prokaryotic cells don't have a mitochondria though, but they still can do this and they're still going to make a lot of ATP. So in prokaryotic cells, this happens on the cell membrane, that outer cell membrane. So in a prokaryotic cell, this happens in the cell membrane, which is still phospholipid bilayer. So in AMP, you're going to learn that this takes place on the inner membrane of the mitochondria. However, it's microbiology, which prokaryotic cells don't have mitochondria, and so this is going to happen out on the outer membrane. Either way, still phospholipid bilayer. You're going to still find all these different enzymes. Again, they're enzymes, they end in aces. Now, it's called the electron transport chain for a reason. All of your NADHs, all your FADH2s are going to come here. This is showing one of them coming here. I think the NADH is showing right there. I know it's right on my line. And they're called electron carriers because they are carrying electrons. And with those electrons, they're also carrying extra hydrogen molecules with them. And so there is energy in those electrons. And when those electron carriers bring that energy, they're going to pump hydrogen molecules across the membrane. So they're going to use the energy of those electrons, and they're going to pump hydrogen molecules across. That's a plus. I'm just drawing a whole bunch of hydrogen molecules. I don't care about a count. So now all of a sudden you're going to have all that energy from electron carriers. And you're going to use all that energy and you're going to pump all the hydrogen molecules that they also had attached to them. Until all of a sudden you have a whole bunch of hydrogen molecules on one side of the membrane. And do things like to be crowded? No, so where are they going to want to go? Right back to the other side. However, they're not going to go through the plasma membrane. They're going to find another enzyme. And they're going to go through this other enzyme. Now, this enzyme, so all these hydrogen molecules are going to go through this enzyme. And the top of this enzyme is designed to spin like a turbine. So when a hydrogen molecule goes through, it spins this like a turbine. Now, this particular enzyme has an important name. It's written up there. But its name is ATP synthase. Again, it ends in ase, it's an enzyme. But it's named synthase because it synthesizes or makes ATP. So its name tells you what it does. Anytime a hydrogen molecule goes through this enzyme and spins the top of that enzyme, again, it's just like a little turbine, like a little wind turbine, it generates energy. And we can take that energy and add a phosphorus onto ADP and make ATP. So we can take that energy and we can take ADP plus a phosphorus and make ATP. So the whole ultimate goal is to take all these electron carriers, use the energy in their electrons to pump hydrogen across a membrane, until all of a sudden you have a whole bunch of hydrogen on one side, and they don't want to be all hanging out on one side. So they're going to then sneak across through this particular enzyme, spin the turbine, and generate ATP. Now I'm going to go back. So I'll come back upwards. So my anything else important. So all the electrons that were in those electron carriers are going to go to the electron transport chain. Eventually they will have to find somewhere to go. And I'm like, I kind of missed that. I'm like, those electrons are going to, that energy is going to help pump the hydrogen. But at some point the electrons do have to go somewhere. And a lot of times those electrons are going to attach themselves to oxygen and they'll make some water. If oxygen is present, if there's an aerobic, if it's an aerobic process. Oops. But the energy's job is to pump the hydrogen ions across the membrane. And then the ATP synthase makes ATP using that energy from the hydrogen ion gradient. Now when I say that, a gradient just means a lot on one side and not a lot on the other side, like a difference. There's a gradient, there's a difference. And they're going to take the fact that there is more on one side, and they want to go where there's not as many, and they're going to take that opportunity to sneak back across through that enzyme, turning the turbine and make ATP. So it makes ATP from the fact that there's more hydrogens on one side than the other. So the where it took place, I already went over. In a prokaryotic cell, outer cell membrane. Eukaryotic cell, the inner membrane of the mitochondria. For what goes in? Well, it's the electron transport chain. And so it's all of our electron carriers. So you almost have to go back on your table and we have to add up the electron carriers that we've generated. How many NADHs have we made along the way? Between glycolysis, acetyl-CoA, and the Krebs cycle. How many NADHs have we made total? We've made 10, because we had 6 here, 2 there, and 2 there. So 10 NADHs go in. And how many FADH2s have we made? Just the two. Just these two. So what goes in are our electron carriers, 10 NADHs, 2 FADH2s. What comes out is ATP. And I'm going to talk on the next little box how much. We get ATP out. We also, I don't think it's showing it on here. If it's an aerobic process, meaning oxygen is present, the hydrogens, because you're still going to have hydrogens hanging out, are going to find some oxygen with the electrons, and they're going to make water. We do get water out. That's all I care about. We do get some water out, because it's H2O. Now, for how many ATP do we make in the electron transport chain? Now, this can vary depending on the organism. Most organisms in the electron transport chain make 34, give or take. On average, 34. Which means ultimately, how many ATP do you make out of the whole process? 38. However, not all organisms do 34. Some are more efficient at pumping hydrogens across, some are not. Sometimes some bacteria have really weak membranes and the hydrogens kind of just sneak through without going through the enzyme, which means then they don't turn it and they don't generate as many. This number can fluctuate anywhere between 30 and 38, or just around 34. But it can change depending on the organism. Now, is it part of the fermentation aerobic or anaerobic respiration? This happens mostly aerobically. If an organism is anaerobic, they can do it anaerobically though. But you guys are aerobic. This would always be aerobic. Some bacteria that are anaerobic can do this. Now, a little bit, at the end of our electron transport chain, we do always have some electrons that have to find somewhere to rest. Electrons don't like to hang out somewhere by themselves. So if there's oxygen present, they will attach themselves to oxygen and make some water. But there's not always oxygen present. Again, that's a horrible picture. So for the electron transport, if it's an aerobic if there's an aerobic process, if it happened aerobically, and oxygen will always serve as the final electron acceptor. And that's where we're going to get water, because the electron's going to attach to oxygen, and it will bring some hydrogens with it, some hydrogens and oxygen, you make water. And this whole process happens more efficiently aerobically. Anaerobic. There are organisms that can do this anaerobically, which means there's no oxygen present, and so those electrons still have to tie themselves to something. So they just tie themselves to any kind of inorganic molecule other than oxygen. What does inorganic mean? They what?
They can't make it themselves.
Close, but you're missing one part of it though. There's no carbon with it. And I'm like, because you're right, they can't make it themselves. And so they're going to tie themselves to some non-carbon, but just not oxygen, because oxygen's not carbon either. In fermentation, because I just wanted to put all the electron where the final electron carriers go. There's always electrons bouncing around. In fermentation, the final electron acceptor is always going to be an organic molecule. So a carbon-based molecule. It's usually going to be some kind of sugar molecule. Let's see. Not going to get through. It's going to be so close. I'm going to have like 10 minutes. I'm going to tape the rest of it because, right, we're done at 10:10. I'll tape the rest of it. It might be like 10 minutes you have to watch on fermentation. Just kind of a couple differences. It's only like 6 more slides left, something like that. And some fun factoids. But I wanted to go over just a reminder on things before I see you next time. I wrote it down. So this PowerPoint will be done. So what do you have due before I see you Thursday? You'll have the little 5-point quiz. Again, you can take them as many times as you want. It will record the higher score. That goes into the grade book. So this quiz will be due Wednesday night before I see you on Thursday. However, I'm like, the next exam, which is going to be on the PowerPoint we finished last Thursday, and this one is due Thursday night. It should be open now. I think so. If you just want to all rush home and quick take the exam. It's due Thursday night. So you'll have a quiz due before I see you Wednesday, and you'll have this due Thursday night. So I'll remind you again on Thursday that you have an exam due, but you don't have to wait until Thursday to take it, because we'll have finished all the information if you want to take it before then. Other things that's not due before I see you next time, but just reminders of things coming up. The next questions about the microbe that you picked are due this Friday. So it has to really do, since we just got done talking about, what are things that organisms need to be happy, certain temperature, certain, substrate elements, whatever, is there a certain pH? What is it that your organisms need to grow? That's what the next questions are based on. So, if you had to grow your particular microbe in the lab, what would you need to make sure you gave it? Like oxygen or no oxygen, or a certain type of cell or something like that. So those are due this Friday if you haven't done them already. Any questions?
Just a reminder about the quiz thing.
Oh, yeah. I'm going to write myself a post-it because I will forget. See? Any other questions? If not, if you already had lab, I will see you Thursday. Email me if you have questions. If you have lab after this, I will see you upstairs.